In order for a cellular organism to survive in nature, it must be able to perform a number of complex tasks, such as acquiring nutrients, assembling a vast array of organic compounds from simple precursors, and ultimately replicate itself in a coordinated fashion. To achieve these tasks, the cell must first separate itself from its surroundings by means of a physical barrier, a cell envelope. Second, the cell may also need to physically separate metabolic processes within the cell by creating intracellular compartments, organelles. The cell surface plays an important role in the biology of a microorganism. The exact structure and composition of the cell surface determines how a microorganism can interact with its external environment, host tissues, and other microorganisms. The properties of the cell surface also determine how well a microorganism can withstand environmental stress in the form of temperature extremes, osmotic or hydrostatic pressures, chemical toxins, and so on. The critical role of the cell surface in the biological function of a microorganism also makes the cell surface an attractive target for antimicrobial therapies, which is why it is worth spending some time taking a closer look at how cell surfaces differ between different groups of microorganisms. A lipid cell membrane is the key distinguishing feature of a cell. The lipid membrane acts as a semi-permeable barrier that enables the cell to maintain a higher solute concentration than the surrounding environment, as well as a stable intracellular pH. The lipid membrane is also used to maintain a membrane potential that is necessary to drive the import of nutrients from the surrounding environment. Since molecules such as sugars and amino acids typically are more concentrated inside the cell, extracellular solutes will not spontaneously diffuse into the cell through cell surface transport proteins. Instead, the cell must use the electrochemical gradient of the membrane potential to drive nutrient import by coupling the import of the solute to the simultaneous import of protons or inorganic ions that are more concentrated outside the cell. Three main categories of cell membrane lipids can be found in nature. Polar lipids, such as dialkyl glycerol diester lipids in bacteria and eukaryotes, and isoprenoid dialkyl glycerol diethers in archaea. Membrane-spanning bipolar lipids, such as glycerol dialkyl glycerol tetraether lipids in archaea. Non-polar polycyclic lipids, such as sterols, hopanoids, and gammacrines in bacteria and eukaryotes. Polar membrane lipids are amphiphatic molecules, meaning that they contain both hydrophilic and hydrophobic moieties. Phospholipids are some of the most common polar lipids in biological membranes. Phosphoglycerides are common in bacterial and eukaryotic membranes and consist of a hydrophilic phosphate group linked to glycerol by an ester bond. The other two hydroxyl groups of the glycerol molecule form ester bonds with carboxyl groups of two fatty acid chains which form the hydrophobic portion of the lipid. The majority of membrane phosphoglycerides also contain a head group, an alcohol linked to the phosphate group through an ester bond. Common head groups include serine, ethanolamine, choline, and inositol. The cell membranes of bacteria that live in phosphorus-limited environments, such as marine cyanobacteria, contain polar sulfolipids rather than phospholipids. Because chloroplasts and other plastids in photosynthetic eukaryotes are derived from cyanobacteria through an endosymbiotic event 1 to 2 billion years ago, plastid membranes also contain sulfolipids. Archaeal membrane phospholipids are significantly different from those of bacteria and eukaryotes. The alkyl chains that form the hydrophobic part of the lipid molecule consist of branched isoprenoids instead of fatty acids. These isoprenoid chains are more resistant to oxidation caused by the extreme conditions under which some archaea grow. Another difference between archaeal phospholipids and those of bacteria and eukaryotes is that the hydrophobic isoprenoid groups are joined to the glycerol backbone by ether linkages rather than ester linkages. The reason behind this difference is thought to be the higher resistance of ether linkages to hydrolysis. Laderanes are some of the most unusual phospholipids found in nature and occur in the membrane of a bacterial organelle known as an anamoxosome. Anamoxosomes are found in a group of bacteria capable of anaerobic ammonia oxidation, a metabolic reaction which takes place exclusively within the anamoxosomes. Anaerobic ammonia oxidation is a slow process, and the unusual cyclobutane and cyclohexane groups within the hydrophobic portions of the laderane lipid is thought to better preserve the anamoxosome membrane potential by preventing leakage of protons into the cytosol. The lipid membranes of some archaea, especially species that inhabit hot environments, contain membrane-spanning bipolar lipids that form highly heat-stable membrane monolayers rather than the typical membrane bilayers found in other cells. These bipolar lipids consist of a cyclic hydrophobic moiety composed of two isoprenoid chains connected to glycerol through ether linkages. The vast majority of eukaryotes, as well as some bacteria, have cell membranes that contain polycyclic lipids such as sterols, hopanoids, and/or gammacrines. Polycyclic membrane lipids regulate membrane fluidity and permeability. Among higher eukaryotes, such as animals, fungi, and plants, polycyclic membrane lipids, primarily sterols, are absolutely essential for proper cell function. The requirement for polycyclic membrane lipids among protists remain to be determined. There have been reports of protists that appear to completely lack any known class of polycyclic membrane lipid. So far, there are no known cases of archaea with polycyclic membrane lipids. Bacteria belonging to the genus Mycoplasma are obligate intracellular parasites that completely lack a cell wall and therefore require sterols to maintain cell integrity. They cannot synthesize these sterols themselves and instead acquire them from the host cell. Sterol biosynthesis requires molecular oxygen, while biosynthesis of hopanoids and gammacrines does not. Therefore, hopanoids and gammacrines tend to be found in microorganisms that inhabit low oxygen or oxygen-free environments. Polycyclic membrane lipids are very chemically stable and can survive for over 1 billion years in the fossil record, which allows paleobiochemists to analyze and classify microscopic fossils as either prokaryotes or early eukaryotes and whether they lived in an oxic or anoxic environment. Although all cells by definition must possess a lipid membrane, a large proportion of bacteria as well as some archaea possess two membranes. Microorganisms with single membranes are classified as monoderm, while those with two membranes are classified as diderm. The compartment between the outer membrane and the inner membrane is known as the periplasmic space or periplasm. Most monoderm bacteria have a thick peptidoglycan cell wall outside the cell membrane that effectively absorbs and retains the crystal violet dye of the Gram stain, which is why these bacteria are commonly known as gram-positive. Diderm bacteria have a thinner peptidoglycan cell wall, which is located within the periplasmic space. The poor penetration of the crystal violet dye through the outer membrane mean that diderm bacteria will instead absorb the safranin dye of the Gram stain and are therefore known as gram-negative bacteria. The outer membrane of gram-negative bacteria is asymmetrical. The outer leaflet of the outer membrane consists primarily of lipopolysaccharides rather than phospholipids, which makes the outer membrane much less permeable to hydrophobic molecules, including many antibiotics. Lipopolysaccharide structures are highly complex and very specific to individual species and strains of gram-negative bacteria. The lipopolysaccharide molecule consists of three distinct components. A glycolipid anchor known as lipid a short core polysaccharide, and an outer polysaccharide containing repeats of an oligosaccharide subunit. Lipopolysaccharide molecules, especially lipid A, are highly immunogenic and can cause endotoxic shock if present in high enough amounts in the body. Ignicoccus hospitalis is a hyperthermophilic archaeon that lives in marine hydrothermal vents. Ignicoccus has a dense cytoplasm surrounded by a periplasmic space that is actually greater in volume than the cytoplasm. Unlike diderm bacteria, Ignicoccus uses its outer membrane for ATP synthesis rather than the cytoplasmic membrane. A cell wall is a common feature of many microbial cells, including nearly all bacteria, all fungi, and many protists. The cell wall helps maintain the shape of the cell and protect it from physical stress, including the danger of cell lysis from the osmotic pressure caused by the generally higher solute concentration inside the microbial cell. The cell wall also serves as a physical barrier against other external hazards such as antimicrobial chemicals or enzymes, as well as parasites. Although the cell wall is designed to be tolerant to physical stress, it must also be highly dynamic so it can be continuously remodeled as microbial cells grow and divide. The microbial cell wall poses a challenge to filamentous microorganisms that need to maintain interconnections between the cytoplasms of neighboring cells. Filamentous cyanobacteria, actinobacteria, and fungi solve this problem by the presence of septal pores. However, the interconnection of cytoplasmic compartments by septal pores makes the microorganism vulnerable to potentially catastrophic cytoplasmic leakage if the cell wall of just a single cell in the filament is ruptured. Consequently, filamentous microorganisms have evolved mechanisms to quickly seal off septal pores adjacent to any cell whose cell wall is damaged. Filamentous fungi belonging to the ascomycetes have specialized organelles called Voronin bodies anchored next to septal pores. If the cell wall of a hyphal compartment is breached, the Voronin bodies of adjacent compartments will immediately plug the septal pores to prevent cytoplasmic leakage. In filamentous cyanobacteria, each end of the septal pore contains both a plug and a cap structure made from protein. The cap structure can adopt both an open and closed conformation and will close within seconds if the surrounding cytoplasm shows signs of stress. Microbial cell walls are primarily composed of polymeric carbohydrates with associated proteins and lipids. The most common cell wall polysaccharides encountered among microorganisms are peptidoglycan, which is a polymer of N-acetylglucosamine and N-acetylmuramic acid and found in nearly all bacteria. Glucan, which is a polymer of glucose and found in all fungi. Cellulose, which is a different polymer of glucose and found in all green and red algae as well as in many protists. Chitin, which is a polymer of N-acetylglucosamine and found in most fungi and some protists. The bacterial cell wall lies exterior to the cytoplasmic membrane and is composed of peptidoglycan, a polysaccharide of alternating units of N-acetylmuramic acid and N-acetylglucosamine. The glycan chains are cross-linked to each other by peptide bridges. These cell wall peptides are synthesized by specialized enzymes instead of ribosomes and often contain unusual non-proteinogenic amino acids such as D amino acids. The peptides are attached to N-acetylmuramic acid N-acetylglucosamine dimers before they are incorporated into glycan chains. Transpeptidase enzymes covalently link the peptides from adjacent glycan chains to create the peptidoglycan network. Since transpeptidases are the target of penicillin, these enzymes are also often referred to as penicillin-binding proteins. This diagram shows the structure and sequence of the peptide crosslink in Escherichia coli. This diagram demonstrates some of the variation in peptidoglycan crosslink sequences that can be observed between different bacterial species. Note the additional interpeptide bridge consisting of 5 glycine residues and the Staphylococcus peptide crosslink. Mycobacteria are a group of monoderm bacteria with atypical cell walls that do not stain with gram dyes. The presence of mycolic acids in the cell wall gives it a waxy property that protects the bacterium from drying out, as well as from bactericidal compounds secreted by host macrophages after phagocytosis. The mycobacterial cell wall is key to the ability of these bacteria to survive in aerosols long enough to infect new hosts, as well as to resist clearance by the host immune system after infection. Molecules are a small group of some 200 known species of bacteria that have completely lost their peptidoglycan cell walls. They are the smallest and simplest free-living and self-replicating forms of cellular life. Molecule genomes have been significantly reduced, and all currently known species of molecules are obligate parasites of animals or plants. The main component of the fungal cell wall is the glucose polysaccharide beta-glucan. Chains of beta-glucan are subsequently covalently cross-linked to each other as well as to other cell wall polysaccharides and structural proteins. The integral cell wall proteins are heavily glycosylated on asparagine and serine residues, predominantly with short mannose oligosaccharides. Some fungi also incorporate the polyphenolic pigment melanin into the cell wall for protection against oxidative stress and harmful radiation. This diagram shows the variation that can be seen in fungal cell wall structure between different species. The majority of described archaea lack carbohydrate cell walls and instead construct an outer protein shell known as an S-layer. The S-layer is a pseudocrystalline two-dimensional lattice made up of one or two self-assembling glycoprotein subunits that are anchored in the cytoplasmic membrane. Many microorganisms secrete an additional layer of polysaccharides over the cell envelope for better attachment to solid substrates or as extra protection against environmental stress or the immune defenses of their host organism. The word glycocalyx is used to refer to bacterial exopolysaccharide layers, which are further classified as either capsules if they are tightly associated with the cell envelope, or slime layers if they are more loosely associated. Many filamentous bacteria living in aquatic environments, such as cyanobacteria, produce specialized protective exopolysaccharide tubes called sheaths. Azotobacter vinelandii is a soil-living bacterium capable of fixing nitrogen at normal oxygen levels, despite the high oxygen sensitivity of the nitrogenase enzyme. To protect the nitrogenase enzyme from inactivation at higher oxygen levels, Azotobacter responds by secreting a thick slime layer consisting of the polysaccharide alginate. Alginate slows down the rate of diffusion of oxygen into the cell to such an extent that biochemical nitrogen fixation can proceed. Many protists construct hard outer shells. Diatoms are a very species-rich group of photosynthetic aquatic protists that construct external shells from silica known as frustules. Coccolithophores are photosynthetic aquatic protists that produce calcium carbonate platelets called coccoliths that are then assembled into an exoskeleton known as a coccosphere. A number of different protists, including foraminifera and some amoebae, construct shells from silica, calcium carbonate, or even sand grains. These shells are commonly known as tests. Prokaryotic cells are generally thought to lack the intracellular compartmentalization typical of eukaryotic cells. However, there are a number of intracellular compartments that do occur in many prokaryotic species that are part of their specific environmental and/or metabolic adaptations. Prokaryotic intracellular compartments are surrounded by either a protein shell or a lipid bilayer. Thylakoids are flattened membrane structures found in cyanobacteria, often stacked in layers that encircle the cell. The thylakoid membranes house both the respiratory and the photosynthetic apparatus in these bacteria. Thylakoid membranes in eukaryotic plastids are of cyanobacterial origin, since plastids are the result of an endosymbiotic event between a primordial non-photosynthetic eukaryote and a cyanobacterium. Chlorosomes are photosynthetic organelles found exclusively among green sulfur bacteria, green non-sulfur bacteria, and Chlorocidobacterium thermophilum. Chlorosomes allow for photosynthesis at very low light intensities. One example of such an environment is 100 meters below the surface in the anoxic waters of the Black Sea, which is home to a stable population of green sulfur bacteria. Gas vesicles are hollow protein structures that are used by some bacteria and archaea to regulate their buoyancy in aquatic environments. A typical gas vesicle is a cylinder-shaped structure that is assembled from a single hydrophobic protein subunit. By regulating the number of gas vesicles within the cell, aquatic prokaryotes can control their vertical position in the water column in response to the availability of light and/or oxygen. Magnetosomes are membrane-bound organelles found in magnetotactic bacteria, a group of bacteria that sense and orient themselves in relation to the magnetic field of the Earth. It is not quite clear what benefit these bacteria get from this ability, but it has been suggested that it can help them determine which way is up and which is down. Magnetosomes are aligned in well-ordered intracellular chains, and each magnetosome contains nano-sized crystals of a magnetic iron mineral. Bacterial microcompartments are protein-bound organelles that typically contain a specific set of metabolic enzymes. Specialized proteins make up the bacterial microcompartment shell, which adopts a symmetrical polyhedral shape. The purpose of the bacterial microcompartments includes increasing local substrate concentration for more efficient enzymatic catalysis, protecting the surrounding cell interior from toxic products, or to prevent escape of volatile metabolic intermediates. Carboxysomes are bacterial microcompartments found in carbon dioxide-fixing bacteria, such as cyanobacteria. The carboxysome typically contains the carbon dioxide-fixing enzymes carbonic anhydrase and rubisco. The function of the carboxysome is to concentrate carbon dioxide to increase the efficiency of rubisco, while possibly also excluding oxygen, which would otherwise decrease the carbon dioxide-fixing efficiency of rubisco. Eukaryotic cells are notable for their complex organization into the intracellular compartments known as organelles. However, there is substantial diversity in intracellular organization among eukaryotic microorganisms that reflect their range of ecological adaptations. This section will look specifically at the structural diversity of three organelles among eukaryotic microorganisms: the mitochondrion, the plastid, and the nucleus. It is currently believed that the common ancestor of all existing eukaryotes possessed mitochondria. Mitochondria are derived from an endosymbiotic event whereby a primordial eukaryotic cell engulfed an aerobic bacterium. Subsequent to the endosymbiotic event, mitochondria in several eukaryotic lineages have begun a reductive evolutionary process as a result of inhabiting low oxygen or oxygen-free environments. Such degenerated mitochondria are collectively referred to as mitochondria-related organelles. Some eukaryotes that inhabit anaerobic environments, or both aerobic and anaerobic environments, have evolved so-called anaerobic mitochondria that use other electron acceptors than oxygen for respiration. For example, some anaerobic protists use organic compounds such as fumarates, which leads to the secretion of succinate as the final product. Hydrogenosomes have lost the mitochondrial genome as well as the oxidative phosphorylation machinery. Eukaryotes with hydrogenosomes instead generate ATP by fermentation of organic compounds with hydrogen gas produced as a byproduct. Eukaryotic microorganisms that possess hydrogenosomes instead of mitochondria tend to inhabit anaerobic or microaerophilic environments such as the digestive tracts of animals. Mitosomes are the most reduced form of mitochondria-related organelle described so far, which completely lack enzymes for oxidative phosphorylation and ATP generation. The only enzymatic property retained from the ancestral mitochondrion is the biosynthetic pathway for iron-sulfur clusters, an essential prosthetic group found in many enzymes. Mitosomes are typically found in obligate parasites which tend to lose metabolic capacity over evolutionary time as a result of their parasitic lifestyle. At present, there is only one known instance of a eukaryote having completely lost all enzymatic functions of mitochondrial origin. The protist Monocercomonoides exilis inhabits the digestive tracts of guinea pigs where it feeds on bacteria. Monocercomonoides belongs to a group of mitosome-containing protists, but Monocercomonoides appears to have lost even the mitosome and the mitochondrial iron-sulfur cluster biosynthetic enzymes by acquiring the equivalent enzymes from bacteria through a horizontal gene transfer event. Plastids refer to a group of organelles that includes chloroplasts, which originated from an endosymbiotic event between 1 and 2 billion years ago, when a cyanobacterium was engulfed by a non-photosynthetic primordial eukaryotic cell. Although the most obvious metabolic property of the plastid is photosynthesis, this organelle also performs additional functions such as biosynthesis of isoprenoids, fatty acids, heme and iron-sulfur clusters. As with mitochondria, there have been several instances of reductive plastid evolution in eukaryotic lineages that have led to the loss of photosynthesis. However, the remaining metabolic pathways associated with plastids have caused them to be retained in many eukaryotic lineages that are no longer photosynthetic. There are a few instances in nature where the cells of eukaryotic microorganisms can contain two genetically distinct nuclei. Dikaryotic cells or dikaryons in higher fungi, micro and macronuclei in ciliates, and nucleomorphs in certain photosynthetic protists. Dikaryotic cells in higher fungi are formed when single cells or hyphae of compatible mating types fuse their cytoplasms but not their nuclei. Fusion of the two nuclei only occurs prior to meiosis, which leads to the subsequent formation of sexual spores. Ciliates are aquatic protists that possess two distinct nuclei with separate functions. The micronucleus is transcriptionally silent during vegetative growth and serves as the repository of the complete genomic sequence and only becomes active during mating. The macronucleus is derived from the micronucleus after mating and undergoes extensive processing, including fragmentation of the chromosomes into short fragments containing just a few genes or even single genes, which can then be amplified tens to tens of thousands of times. Nucleomorphs are vestigial nuclei containing shrunken genomes and are the result of secondary endosymbiotic events, where a non-photosynthetic eukaryote has engulfed a photosynthetic eukaryote, either a green or red alga. All photosynthetic protists are descended from secondary endosymbiotic events, but the nuclei of the engulfed algae have only persisted as nucleomorphs in the cryptophyte and chlorarachniophyte lineages. The engulfed algal cell has formed a distinct intracellular compartment known as the periplastid compartment. Nucleomorph genomes encode essential plastid genes that cannot be transferred to the nucleus, which explains why nucleomorphs have persisted in these lineages.